Uh, a very good morning to all of you. Today I am going to discuss about the cell division. Okay, so just see in cell division topic, the division of the cell. So division of the cell include two events. One is division of uh, nucleus. So if the division of nucleus involved, then it is known as karyokinesis. Division of nucleus, it is known as karyokinesis. But division of cytoplasm, that is known as cytokinesis. Karyon means nucleus. So if division of nucleus occur, then it is known as karyokinesis. But if the division of cytoplasm is occur outside the nucleus, means we are going to get the two different identical cell, that is due to division of cytoplasm. But we cannot expect two identical cell just after division of karyon, that is known as nucleus. So after two events we get two identical cell. That two events is karyokinesis and other is cytokinesis. So if anybody asks what is cell division, that is the division of nucleus as well as division of cytoplasm. So in case of division of nucleus, there is equal distribution of chromosome and equal distribution of nucleoplasm. So that involves to get the two nucleus. But in case of cytokinesis, just after karyokinesis, there is cytokinesis occur. We cannot expect cytokinesis before karyokinesis. So it is visible to all of you. It is visible to all of you. Okay. Yes. So just try to know. Cell division, that is three types, mainly amitosis, that is occur in prokaryotic cell. The amitosis, that is occur in prokaryotic cell. So that is the bacteria. But mitosis, that is occur in somatic cell. Mitosis occur in somatic cell. I have already discussed these things. What is somatic cell? That is responsible for growth and development, that is responsible for repair. And you can observe this somatic cell in case of plant inside the leaf, inside the root, inside the stem, and all the part of the body of the plant except inside the flower, where there is a formation of male gamete and female gamete occurs. So second type of division, that is known as meiosis. Second type of division, that is known as meiosis. So where meiosis occur? Meiosis occur in reproductive cell. Meiosis occur in reproductive cell. Okay. So are you observing? It is clear to all of you. Meiosis generally occur in reproductive cell. Okay. So what is reproductive cell? That is the germ cell. GRM. Germ cell. Other name of reproductive cell, that is known as germ cell. Okay. So generally male and female reproductive organs, male and female gonads, gonads, gonads. That is in case of animals. So there is a formation of male gamete, that is known as sperm, and the formation of female gamete, that is known as egg. So spermatogenesis and oogenesis, formation of male gamete and female gamete, that the process, that is spermatogenesis, and then it is known as oogenesis. So that is responsible for making of the sperm, sperm, that is male gamete. It is known as male gamete. Okay. And This is oogenesis is responsible for formation of egg. So generally, reproductive organ is your reproductive part of the body, that is flower in case of plant, but in case of uh, animal it is separate. So the animal that is dioecious. Okay, dioecious means male and female part will be there. Male part plant, male animals and female animals like that. But in case of plant, it is bisexual flower. Sometimes you will find the male plant and female plant, like in papaya. Okay, so in case of papaya, there is a male plant and female plant. So meiosis generally occur in reproductive part, not in all the part of the plant of body, or not all the part of the animal or human body. Only a specific area that is localized for formation of either male gamete or female gamete. So male gamete, that is a sperm, and female gamete, that is egg. Female gamete. Female gamete. Okay. So all of you, it is clear to all of you? It is clear to all of you? It is visible or not? Yes.
Sir,
Uh, Ashish, it is clear?
sir,
Yes. Yes. Me. It is clear. Yes. So
Yes,
now it is
clear.
clear.
Yes, sir. Very clear.
Okay. So just I'm discussing. I'm going to revise the cell division, that is division of carrion, that is nucleus. Karyokinesis, that is division of nucleus. Division. Division of nucleus. Okay. And then other is division of cytoplasm. Division of cytoplasm. Cytoplasm. Okay. So first event of the cell division, that is division of nucleus, that is carrion. Carrions means nucleus, and division of cytoplasm, that is the liquid part outside the nucleus. That is, we are going to get the two identical cells just after two events. First event, it is karyokinesis. Then second event, that is cytokinesis. Without cytokinesis, we cannot get two equal size cells after cell division. So both events are equally important for getting the two daughter cell or four daughter cell. In case of mitosis, we get two daughter cell. In case of meiosis, we get four daughter cell. Okay. So I have discussed that is bacteria, that is prokaryote, inside which there is a mitosis occur. A mitosis, that is simply occur in case of somatic cell, and meiosis generally occur in reproductive cell. So you know the reproductive cell. What are the reproductive cell? That is the male gonads and female gonads. Male gonad, male reproductive organ, and female reproductive organ. So inside that there is a formation of the male gamete and female gametes. So what process is going on? That is meiosis. Okay. So I have already told you mitosis is equational division. Equational division. Okay. And this meiosis is known as reductional division. Meiosis is known as reductional division. That is reductional division. Okay. So you, all of you know what do you mean by equational division? Means same number of chromosome will be there inside the daughter cell just after cell division, if you compare with the parental cell. If you compare the mother cell or parental cell and daughter cell chromosome number, same number of chromosome is there in case of mitosis. But in case of meiosis, the chromosome number is going to reduce. That is N. N means haploid. Haploid. N means haploid. But in case of mitosis, there is a 2N. N means diploid. N means diploid. So same number of chromosome, if it is there, then it is known as diploid condition, and if the half number of chromosome is there in uh, daughter cell, then the process is going that whatever you get the haploid only after meiosis, not after mitosis. So this is the difference between mitosis and meiosis. Meiosis is responsible for maintaining the chromosomal balance. You know, the 2n, suppose 2n number of chromosomes present in the reproductive organ, but the male gamete and female gamete is only haploid. Reproductive organ cell have 2n, but the male gamete form, female gamete form, that is we get the n. Suppose it is coming from the male, and then 2n chromosome number, that is the cell of the reproductive organ where 2n is there, but whatever it is formed, that is haploid. So you get the haploid, that is n plus n. So male, male, you indicate like that, male like that, and female like plus. So just this is the symbol of male, this is the symbol of female, and you are getting the haploid chromosome number. Okay. So this process just going to reduce the half. 2n is equal to double of n. That's why we are telling that this is the reductional division. That's why we are telling that this is the reductional division. What is 2n? 2n is the number of chromosome, that is the double in number. Okay. So n means haploid set of chromosome, 2n is diploid set of chromosome. So that is generally the somatic cell, the chromosome number remain same, that is 2n, that is diploid, but reproductive cell, the reproductive cell, just after process of meiosis process, we are getting n inside the nucleus, that is haploid in number. So now come to the meiosis. Meiosis, there is a two part. So meiosis, there is a two part. That is, you will get meiosis, that is meiosis 1. This is meiosis 1, and this is meiosis 2. This is meiosis 1, that is reductional division. Actual reductional division, we can say meiosis 1. Actual meiosis is meiosis 1, but meiosis 2 is just like mitosis. Meiosis 2 is just like mitosis, but included inside the complete the process of meiosis. So what is the meiosis 1 means? You will get the different stage of meiosis, that is prophase 1, that is metaphase 1, anaphase 1, and telophase 1. It is clear to all of you? Means you are getting the five different stages, that is four different stages: prophase 1, metaphase 1, anaphase 1, telophase 1. And then we proceed for cytokinesis. After that, cytokinesis, for division of cytoplasm. Okay. So after that we are going to proceed for division of the cell in two equal parts. Okay. Now just to see meiosis 1, that is prophase 1. So prophase 1 is the longest stage of meiosis 1. Okay. Longest stage, longest stage of meiosis 1. Why? Because you get the five different sub stages of prophase 1. You get the five different sub stages of prophase 1. That is leptotin, zygotin, pacatin, diplotin, and dikinesis. So leptonema, you can also see the word leptonema or leptotin, both are same. Then zygotin or zygonema, both are same. Then pacatin or pacanema, that is both are same. Then diplotin or diplonema, that is both are same. And dikinesis. So these are the five different sub stages of meiosis prophase 1. So this is the longest stage in case of meiosis, cell division. And then the cell will enter in metaphase 1. Then cell will enter into of metaphase 1. After completion of metaphase 1, the cell enter into anaphase 1. Okay, and then telophase 1. Then there is a division of cytoplasm. After division of nucleus, you get the division of cytoplasm. So you will get two equal cells. Further from this two, again there is a formation of two. Okay, then there is a formation of two. Where in the case of meiosis two. So from one cell you get the two equal cell, that is haploid cell from one cell, and second you also get two equal cells. So in complete process of meiosis you will get four different haploid cells, but in case of mitosis you will get only two diploid cells. You know the word diploid and haploid. Means in case of mitosis we are getting the 2n condition of the chromosome inside the nucleus, but in case of meiosis we get the n condition, haploid condition of the chromosome inside the nucleus. So you are getting only two cell in case of mitosis, but in case of meiosis we are getting four haploid cell. Instead of two we are getting four haploid cell. How? Means first the meiosis one will produce two cell, that is haploid cell. Now these two haploid cell again first further divide and get the two in number from one haploid cell, again two in number in second haploid cell. Okay, so this is the process, that is the meiosis 1 and meiosis 2, that is completely different. Meiosis 2 just like mitosis process, what you are observed we have seen in mitosis. It is clear to all of you? It is clear to all of you? Yes. Atharva? Yes. uh, uh,
Yes, sir.
yes. So can you tell me that what are different sub stage of meiosis? What are different sub stage of meiosis? Yes. Reply. Meiosis 1 and second meiosis
Sir,
2.
metaphase.
No, what are different sub stage of meiosis? What are different sub stage of meiosis? That is meiosis 1 and meiosis 2. So what are the further sub stage of meiosis 1? What are the further sub stage of
Prophase.
meiosis 2? Prophase 1, metaphase 1, anaphase 1, telophase 1. Can you tell me the further sub stage of prophase 1? Yes. What are the further sub stage of prophase 1?
Sir, lipotine.
Leptotene. Leptotene. Then
Sir,
zygotine. Then
नहीं सर मैंने
zygotine. Then pectine. Then diplotene. Then diakinesis. Okay. Now
yes,
liptotene is
sir.
also known as liptonema. Zygotine is also known as zygonema. Pectine is also known as pecanema, and diplotene is also known as diplonema. Okay, diplonema and diplotene both are same. Diagnosis and diagnosis. Okay, so these are the sub stage. This is the longest stage in case of meiosis, that is prophase 1. So that have 5 further substages. So if anybody asks can you bifurcate the meiosis, then you will tell you that there is a meiosis 1 and meiosis 2. In case of meiosis 1 there is a prophase 1, metaphase 1, anaphase 1 and telophase. There is further substage of prophase 1, that is leptotene, zygotine, diplotene, leptotene, zygotine, pectine, diplotene and diakinase. Then the cell will enter in metaphase 1, then cell will enter in anaphase 1, then cell will enter in telophase 1. Then the process will start, that is cytokinesis. Without cytokinesis, the cell will not proceed for meiosis 2. So first event is meiosis 1. In the meiosis 1, first event is prophase 1. In prophase 1, there is a cell will enter in leptotene, then cell will enter in zygotine, and then cell will enter in packaging, and then cell will enter in diplotin and then diagonose. After that, it will enter into metaphase 1, then anaphase 1, then telophase 1. Then there will be cytokinesis occur. After that, it will enter into meiosis 2. And this meiosis 2 is just like mitosis, that is prophase 2, metaphase 2, anaphase 2, telophase 2. Then what is the difference between meiosis 1 and meiosis 2? This is the question mark. So in case of meiosis 1, you know that they are 2n is equal to suppose example in case of human being 46. So they chromosome division occurs. Means first the cell will get equal number of chromosomes. Two daughter cell will get equal number of chromosome. But in case of meiosis 2, there is a chromatid separation occur. In case of meiosis 1, there is a final chromosome separation occur. But in case of meiosis 2, there is a chromatid separation occur. So you are familiar with chromosome, chromatid, centromere? You are familiar or not? You know the word chromosome, chromatid? If you don't know, then I will explain. Are you
सर
familiar?
बताया था आपने पहले.
हाँ बताया था. So each chromosome is made up of. I'm just erasing that portion. Okay. So each chromosome is made up of two chromatids. Each chromosome is made up of two chromatids. Okay. So just see what is happening in case of meiosis 1. Okay. So just I'm starting the meiosis 1. That is prophase 1. This is the first stage. In prophase 1, you will find further substage. Okay. So this that is meiosis. I'm starting meiosis. That is meiosis 1. That is in meiosis 1 I'm explaining prophase 1. Now in prophase 1 I'm going to explain the leptotin. Leptotin. So what is leptotin? So suppose in case of leptotin, what you will observe, suppose it is a nucleus. Okay. So if it is early stage of leptotin, so what happens? You will observe the thin thread-like structure of chromosome that is known as chromatin, and this is the cell. This is the cell structure. Okay. So generally you will find the in case of leptotin you will find the this is the nucleolus. Okay. And this is chromatin, and this is the nuclear membrane, and you will find this is the cytoplasm like that. It is the cytoplasm like that. So this is the very early stage. Now what happens in leptotin? In case of zygotin, what happens? The chromosome start to become condensed, means thicker. Okay, so you can observe the this one is the chromatin. This is chromatin. So what is chromatin? Thread, thread-like structure, thread-like structure of chromosome. Chromosome. So this is the first stage of meiosis 1, that is prophase 1. And you observe the nuclear membrane. You will observe the nuclear membrane. Okay. And it is very thin, thin chromatin and thread-like structure. Inside the nucleus, you will find the nucleolus and nuclear membrane. This is the cytoplasm. And this stage is known as leptotin. So now this process will start only inside the reproductive organ. Reproductive organ, that is the flower. Okay. So that is the testis or ovary. You know testis or ovary. So that is the male reproductive organ, that is the female reproductive organ. Okay. So in case of plant, male reproductive organ part and female reproductive organ part. So now the next stage, that is known as zygote, means chromosome have become condensed. This thread like a structure, it become condensed. What do you mean by condensed? Means you can observe what you can observe. It is clear to all of you, leptotin is clear or not?
Yes, sir.
Yes. So if thread-like structure or nucleolus is present, a nuclear membrane is present, then this stage is known as leptotin. Next, I am talking about zygote. Next, I am talking about zygote. What is zygote? The chromatin thread-like structure become condensed. Chromatin thread-like structure become condensed. So what you observe? You can observe like that. You can observe like that. This is the bivalent chromosome. You can observe in case of zygote. Okay. So you can observe like that. Okay. So just see, you will observe this thing. This is the chromosome shape appear. Means initially it was thread-like structure. Now it become that is thick chromosome-like structure like that. If you see in the enlarged view, then you will say it is a chromosome. It is a this is a chromatid. Okay, this is a chromatid, and you will find this is the centromere. This is the centromere. This is one is the centromere. Okay, and this is the chromatids. Means this is A and this is B. A and B both are chromatids. This is a chromatids. Chromatid. Okay, so two chromatids make a chromosome, and that is joined together by the regions that is known as centromere. So that same chromatin is going to condense and become three. Okay, so that you can observe in case of zygote. What is this? ZY zygote means, and you can observe there is a pairing, means bivalent, means two chromosome come together. So you observe the two chromosome like that. Two chromosome like that means, suppose this is one chromosome and this is the second chromosome. This is the second chromosome. Okay, and you will find this is the second chromosome. So this one is known as bivalent. This one is known as bivalent. Bivalent. Bivalent chromosome means whatever the materials, a protein material like that, you will find materials between these two, that is synoptimal complex, synaptonymal complex, help in pairing of two chromosome. Help in pairing of two chromosome means there are four chromatin. If this two chromosome is coming closer with the help of synoptimal complex, then pairing, that is known as bivalent structure you can observe in case of zygote. Bivalent structure you can observe in case of zygote. So this two chromosome is going to complex, means come close together, and bivalent structure form. So what is the advantage of this bivalent structure? Just see, this is that means, just after zygote, this there is a crossing over occur. Just after zygote there is a crossing over occur. What happens in crossing over? You will find like that. You will find like that, means, there is a exchange of chromatids occur. There is exchange of chromatids occur. So I am going to explain now, that is the pacatin, that is the pacatin. Okay, so this stage that is known as pacatin. So what happens in case of pacatin? You can observe with the help of this diagram. What happens in case of pacatin? You can observe with the help of this diagram. Okay, so what is happening means there is a bivalent of two chromosome means four chromatids, because one chromosome is made up of two chromatids. This is the one two chromatids of one chromosome. This is the whole one chromosome. Okay, and this is whole one chromosome because two chromatids constitute together and make a chromosome, and this is the centromere of two different chromosomes. Okay, now the exchange between two chromatic cross-like structure. You see the cross-like structure that is known as chiasmata. So this stage I am explaining that is known as pachytene. Pachytene, pachytene. What is happening in case of pachytene? What is happening in case of pachytene? There is exchange of segment of chromatids. So what do you mean by chromatid? The two part of the chromosome that is known as chromatid. So this is the both are the similar. That's why this is known as sister chromatid. This is known as sister chromatid. This is known as sister chromatid. But this is known as non-sister chromatid. These are the chromatid of two different chromosomes. These are the chromatid of two different chromosomes. This chromatid is of this chromosome and this chromatid is of this chromosome. This chromosome. Okay. So what happens in case of pachytene? Exchange of segment of the chromatids of non-sister. What do you mean by non-sister? This is non-sister for this chromosome. This is non-sister for this chromosome because this is not a part of this chromosome, and this is not a part of this chromosome. That's why we can say it is the non-sister chromatids for each other. So what happens? What happens? There is a crossing over occur. Cross shape structure that is known as crossing over. Crossing over occur. Crossing over. Crossing over. Cross shape structure, that is the cross shape structure that is known as crossing over. Crossing over. And what happens? That is a cut occur. After crossing over, cut occur. And again joining of that chromosome occur. Again joining of that chromosome occur. So what happens? This is going to separate, and now this is going to separate. So that is the main reason for variation. That is the main reason for variation. If you are if you are two brothers, and if you have brother and sister, okay, even then you are not similar. Why? Due to this crossing over. And that is the main reason that due to crossing over, there is a segment of variation of the segment of the gene. Means that gene is present initially in that chromosome, and suppose there is a gene that is present initially in that part of the chromosome, but now it is become part of this. So you will get the variation means some part of segment of this chromosome is there in this chromosome, and some part of segment of this chromosome is there in this chromosome. If because you see what happening if you are going to separate लेफ्ट अपवर्ड एंड राइटवर्ड डायरेक्शन, अगर आप इसको सेपरेट करेंगे इधर से और इधर से इस लाइन के थ्रू, देन यू कैन ऑब्जर्व दिस सेगमेंट वाज नॉट इनिशियली दैट पार्ट ऑफ क्रोमोसोम, बट नाउ इट इज द पार्ट. Means the gene present in that region, it is come along with that chromosome, and gene present in that region, it was the part of that chromosome, now it is the part of that chromosome. So it causes a variation. So due to that variation, you can observe the difference variation between the two sisters, variation between the two daughters, variation between the four daughters, or variation between the four brothers and four sisters. So this is the main reason, that is crossing over. So this meiosis, that is the mystery, that due to that crossing over you can get the variation. So that process occur in case of pachytene of meiosis 1. So this one the third stage of the meiosis 1, that is the first stage is leptotin, second is zygotin, third is pachytene. Now what happens after getting such type of chromosome, that is the separation of that cross shape structure, that occur in case of diplotin. So after diplotin you will get such type. just cross shape structure formation, that is the stage of packaging. And just after getting such type of structure, that is occur in case of zygote, that is that is terminalization of crossing over, means end of the crossing over occur in case of zygote. And then you get the diagnosis, means finally the chromosome that contains some part of the other chromosome, and the other chromosome that contains some part of the first chromosome. So you observe in case of diagnosis. So whole process of the prophase 1 is going to complete. Now what happens? All the chromosome will start moving toward the center. All the chromosome in pair. It will not come in single. It will come in pair. All chromosome just like that. All chromosome of your body cell that will come toward the center in case of metaphase 1. So this is the difference in metaphase 1 and metaphase 2. What happens? In case of metaphase 1, the two pair of chromosome arrange, but in case of mitosis, only one chromosome layer is going to form. So this is the major difference in metaphase 1 and mitotic metaphase, or metaphase 2. Mitotic metaphase and metaphase 2 is the both are same, but meiotic metaphase it is different, because two layer of chromosome you can observe in the center of the cell in case of meiotic metaphase. But in case of mitotic metaphase or metaphase 2, only one chromosome layer is. This is the major difference in metaphase 1 and metaphase 2 or mitotic metaphase. It is clear to all of you? I will explain on next day. Okay. So thank you very much. If you have any question, you can ask to me. कोई क्वेश्चन है?